Good morning, everybody. Mr. Chairman, Professor Shamon, Professor Hashinawi, thank you very much for your kind invitation. Unfortunately, I prepared a preparation in French and I'm faced now with one in English. I do have another one in French, but I think that it's not suitable for the audience. So I'm going to talk about the blood banking in general, just giving an overview about the situation, the actual one in Egypt, in the Middle East especially, and in the whole world. I consider the stem cells in the blood really as the seeds of hope. As you all know, it's the most important today for us as source for stem cells is the blood, adipose tissue, bone marrow, but the most important we are going to stress on the blood. Banking the blood is a way of preserving potentially life-saving cells, which are the stem cells of hope that usually get grown after birth anyway. Nowadays, we are not talking anymore about only hematopoietic, but because of the stem cell plasticity, they can be transformed whether into somatic cells and vice versa, the hematopoietic ones. Some numbers regarding what's going on in the world nowadays. As you all know, of course, they are immature, these cells, and are very important for this reason. We do have more than 20,000 reported blood transplant worldwide. More than, for example, in the USA, more than 20 bloods operating in the United States of America since 1992. And today, maybe as you said, today more than 500 or more than 400, let us say 500 blood grafts available in the world in more than 50 blood banks worldwide. For example, in Japan, approximately 50% of hematopoietic stem cells from unrelated donors are being performed from the blood cells. And this, I am very interested in this website, please have a note, because if you go into this website, you can know a lot about the blood banking at Unit or Network, for example. As an overview about the blood banking in the world, it's whether private for family banking or public for donation. And the most important for us, of course, is the public ones. What about the blood banking in the Middle East? I have been faced a few days ago I have been really astonished to know that nowadays in Jordan, it is, unfortunately, it's not in this lecture, in the French one, I wrote it, that in Jordan, now there are 6 blood banking operating in Jordan. They are only, as population, only 5 million. We are in Egypt, 85 million. And we do not have, we have just one private. So in the United Arab Emirates, they do have one started in 2001. In Iran, for example, I think they do have more than one. Jordan, as I said, 6, astonishing really. In Israel, nearly 5 nowadays. And if we talk about the store, how is the product is stored, it either in the form, let us say, nucleated cells or mononuclear cells, or some banks are storing the blood as whole blood, which is really very interesting. At the German, some in German, and another one in Denmark. The stored blood are either in bags or cryovials to save this, and these blood banks should be accredited by the American Association for Blood Banks. Just an idea about the fees in the private ones, of course, the initial fees are around $2,000 to store the blood, $2,000 US, and yearly for $150 for up to, let us say, 20 or 25 years. So what about Egypt? Egypt, we do have a private bank, it's called CellSafe, and this one started in 2008, and they do have, actually, I just lectures, you have to keep in mind that the most important testing for any cord blood unit are the nucleated cell counts, the microbiological testing. Of course, we have to screen for any viruses like CB, HIV, and of course we have to preserve viable cells all the time, so we have to test for viability. Optional, in my opinion, for what's going in the world, it's important, but at the same time they consider it optional, actually typing, and sometimes we do really need high resolution, and sometimes we need genetic screening. We all know the potential, of course, uses of the stem cells, but the most important, of course, nowadays is to use either in bone marrow transplantation or in regenerative medicine. And this is a statement from Madame Blukman in Hematologica 2009 that cord blood is an unlimited source of hematopoietic stem cells for allogeneic stem cell transplantation. The regenerative medicine, of course, is becoming a very important field of application for the stem cells, and especially the stem cells found in cord blood. But in my opinion, it must not put an additional load on our budget as developing countries, and should be utilized to improve the global health equity rather than to contribute further to the problem. It should provide permanent solution and reduce the financial burden, of course. And this, I like it, because when we are talking about the WHO, we are all thinking of the infectious diseases, but unfortunately it has been published by the WHO that non-communicable diseases like diabetes, chronic vascular or cardiovascular diseases, and cancer, that all the neglected epidemics account for more deaths annually than infectious diseases. And by the year 2020, it is estimated that 7 out of 10 deaths in developing countries will be attributable to non-communicable diseases. So we have to look for a source of, life or saving for those diseases like those stem cells. So why are we thinking about the cord blood stem cells? What the impact or the value of these cord blood stem cells? They are collected via non-invasive procedure, very easy to obtain. Cord blood transplants result in lower incidence of life-threatening side effects, such as the graft-versus-host disease, occurring frequently a little bit more with EMT. I'm saying less, not at all. And less latent infections. Why? Because cord blood stem cells are somehow, as we have seen in the previous very valuable lectures, they are immunologically immature or naive, giving them better chance for being accepted. I like very much this slide, and all the time stressing on it, because you have to take a home message regarding what's stem cells, what are the compartments of fractions of cord blood, let us call regenerative cells or stem cells. You have the hematopoietic, if we fractionate the whole spectrum of stem cells found in cord blood, the hematopoietic compartment, the endothelial angiogenic one, the mesenchymal one, I consider it the most important one, and the unrestricted somatic, which is somehow similar, somehow has some properties or some similarities with the embryonic ones. Of course, the cells released in the bone marrow or in the cord blood are adult things. They do not have anything to do with the embryonic stem cells. Just a few words about the mesenchymal fraction. The mesenchymal fraction is a fraction that this fraction doesn't express CD34. It's negative for CD34 by immunophenotyping, also negative for CD45, HLA-DR, but positive for STRO1, CD28, 44, and 51. The most important to separate these mesenchymal stem cells, they do have the function to adhere to plastic surfaces, so easily isolated in culture flasks, and they do possess a fibroblastoid morphology. They do have a high expansion potential and, of course, some extended plasticity potential. Others, they are somehow, they do have a low immunogenicity, considered as immunomodulators, and anti-inflammatory. They also have some anti-inflammatory properties. They secrete some immune inhibitory cytokines like interleukin-10, transforming growth factor beta, and we can program that, or we can consider they have tolerogenic epidemics. Who will benefit from these stem cells in the poor blood? Most importantly, the children. Just to keep in mind that the transplantable dose that we use in poor blood, not like in bone marrow, it's the nucleated cells. And the average dose is around 3 10 to the power 7 nucleated cells, and we measure it like in bone marrow transplantation according to the body weight. Okay, and in adults we have to consider two important issues. The surface area, of course, of the adult is bigger, so we may need double coats, of course, but here we have to consider action in matching, because between the two coats we can accept up to around 1 action in mismatch. Published by Madame Beauchamp, the colleague of Professor Madame Gluckman in 2001, that Eurocode published a study comparing the outcome of match. It's somehow old, this slide. The outcome of matched unrelated bone marrow transplant. What does it mean, matched? It means that actually 6 out of 6, okay, either unmanipulated or T-cell depleted to mismatch umbilical cord blood transplant. What does it mean? That actually 2 out of 6 mismatch, the results were found to be similar. Who are the main players in this story? Of course, the producers. If you are talking about cord blood banking back, the producers are the OBG or the obstetric units, and of course high-tech laboratories to preserve or collect or process the stem cells in the cord blood, and the end users, whether they're transplanters or researchers. In Egypt, for us, the transplanters are the most important, because we do have since a long time a transplant team headed by the president of Cairo University, Professor Kassem, and they are very successful in it. The project standards, I don't know if it is interesting for you, but anyhow, I like to say that this project, as an industrial project or somehow in the world, we have to comply to the international standards. We are not working on ourselves, so we have to bear in mind some terms that are very important, like that the project should comply to the good manufacturing procedures. Sometimes you read it, GMPs, meaning good manufacturing procedures, should comply to the current good tissue practice, and the EU standards, of course, or the European standards, they began a long time before us, so we have to follow their steps. We do have in Egypt an Egyptian code for practice for tissue banking. I like very much that you go and learn about it. It's an Egyptian one. It covers so many important points, and we have to learn it. It is also called the MIHRA, or Medicine and Healthcare Products Regulator. Regarding the processing, this is a very important point in the processing of the cord blood samples in the bank. We have to look for preventing the disease transmission, maintain the function and integrity of cells, standardize a closed system and just to know that each for the blood unit should be overlapped. This is very important, I mean, from the technical point of view, with plastic prior to storage, because sometimes you have very important problems regarding the cryopreservation and some failure of some samples. These are the, I like to finish with this, especially in Egypt, to call for teamwork, and as Truman said, that it's possible to achieve anything as long as you are not worried about who gets the credit. Thank you very much.
Thank you to Dr. Neamatt for the interest and scientific thesis lecture. Any questions, please?
Of the endothelial cells, you mean that what has been done in Egypt or in general? In general, we didn't try in Egypt, but in my opinion, this is very important for the vascular problem, the angiogenic, and people with limbic or limb ischemia. And there is a team in Egypt, they tried something like that, but on a very small scale. It has not been published, but as it is clear, it has to go very well with the, I think, with the limb ischemia.
But I think in Egypt what done is not leukemia, it's in kind of stem cell.
Yes.
For the injection, for the vascular cancer or something like that.
Yes, they are somehow comparable. Myself, I worked on some mesenchymal stem cells regarding the laboratory, but not, I mean, the application. If it is mesenchymal or endothelial, as we have said this, as you have seen, it's just a fraction of stem cells, but the mesenchymal, in the beginning, as it is somehow not very immunogenic, doesn't express CD34. Maybe the mesenchymal can be acceptable more.
I know the vascular surgeon
Yes.
do that. It was good for Hannah.
With the mesenchymal one.
Exactly.
Yeah, but what I mean that in Egypt we cannot go into clinical trials for the moment because we are still in the research level. We need a long time to go for clinical trials and publications and so on because it's a human being. It's not uneven.
But as for this little thing, I think this is published internationally. It is later as
I don't know, they published, I think not. Yeah. They published on the cells in the angiogenic potential.
For vascular ulcers and resistant ulcers.
Yes, what's the superficial? Yes.
This was a very nice and very informative presentation. I want to actually ask you a question which I don't think Virginia either spoke about that. A few years ago there was a lot of hope about in vitro amplification, and I don't hear much about it at the moment. Do you have any recent information about actually expanding the cord blood cells either prior to storage or prior to transplantation?
About expansion.
Expansion, yes.
In Egypt or...
Well, what you know, I don't know anything about at the moment.
I think there have been trials, especially in France. They have been very enthusiastic about a few years ago about the mesenchymal fraction of cord blood, and there has been a team culturing and extending the mesenchymal stem cells and using them as a treatment for GVHD. Yes? And then I don't know why did they stop. I don't know the reason really.
stopped because there was actually some induction of
Immunogenicity,
tumor-like
yes, yes.
information. So
Yes.
that's been resumed. But that's what the mesenchymal said. I was mostly thinking of expansion of the hematopoietic progenitor as a whole.
Yeah.
Because the question was basically, if you expand, you may actually differentiate. And that conundrum has not been fully worked out. So,
Yes.
I haven't followed the literature in the last 2 years, but I don't think I've seen.
Yes, in my opinion, I'm sorry.
Yeah, I'm asking your opinion.
In my opinion, if we induce the cells to differentiate, or I go into culture and subcultures and so on, I'm pushing the cells to differentiate, and this is not the right way.
Yeah,
Because they, by the end, they will die when I inject them.
I share your opinion.
So it's better to get the benefit of the few months at the beginning when they are immature or somehow naive, and then they will start to differentiate somehow in the body in vivo, and then afterwards we are going to need a booster dose. It's something like vaccine, or because they do have a lifespan.
The second thing, if I may, you mentioned very positively the situation in Jordan, where there are 6 full-blood banks, which I think they should be congratulated. But the question is, how did they achieve that? And maybe we should, and after that, try to understand how they achieve that, then whether this is applicable or not. I don't know.
I have been astonished, really. I learned this these data a few days ago from a Jordanian who was attending a conference with us. I do not know. They are only
Because information on corporate banking is not that easy and that transparent. I mean, to try to know exactly what are those corporate banks and where they are and who controls them, are they real or not real? It's not an easy task.
Yes, are they standardized? Are they accredited? What's going on?
Do you know how they're private and public?
I think it's a very important issue. I like this point because, in my opinion, the blood banks should be either public or semi-public. But most of them, I think, in Jordan, most of them are private. I think.
That's a debate we
Yes, it's a very big debate.
could have. I would agree that they should be all accredited.
Yes.
That's for sure.
Yeah.
They should be devoted to either allogenic or autologous.
Yes.
Just to say they should be private or public, they should be controlled by public regulation.
Yes.
Unfortunately, I think it's the case in France, it's the case in Egypt, it's the case in many places. Public system doesn't work all very well.
Yes.
So I think we should not claim too much. It has to be public. It has to be for public use. That's a very important thing. But if it's run in the private system, but if it's controlled, this is all the pharma, all the drugs we are using for patients are actually private companies. They are controlled, and that's beneficial. I don't see why there should be such a difference.
I do have some, Professor Sharon, I do
Yes.
have
Merci.
some
En
information
français.
about this.
But maybe I
And
can.
the French one, because this, I collected some more information, but because this is a very important issue that we have to go for the public ones. And now in Egypt, we do have a code of ethics regarding the code of blood banks, regarding what we call in English the triple P, or the public-private partnership. It should not go on its own.
No, it should be regulated.
Yes,
I agree.
So,
But maybe I can get it on a key, so I
yes,
can read it.
please.
I'd be very pleased to get
okay,
that.
thank you. Thank you.
Any questions? Thank you.
Thank you.
Also,
some mismatches only, because we are talking about, if we take 6 as our cutoff of as a figure, if you are going for all the blood, let us do it in bone marrow. Stem cells from bone marrow, better to be 100% matching T-cell depleted. And if it is all the blood, we can accept up to 2 mismatch, 2 out of 6. Yes, mismatch 2. But if you are using 2 cord bloods, 1. Better, because you will have, because we have to consider the HLA, we have the major and the minor. Nobody is talking about the minor. You have problems from many other issues. The preform, the antibodies, the minor HLA, and so on. We are trying to minimize. So if we are using 2, only 1 out of 6. Yes, it's very important. Yes, yes. No, it's very important. But what I meant is that, for example, in the family banks that we do have in Egypt, they do not make anything regarding the HLA. They are just putting on the label, yes,